Hi everyone, Dr. Rashid here. In this video, I will be explaining you the structure of RNA. So regarding RNA molecule, it is a polymer of ribonucleotide. And RNA contains only a single strand of this ribonucleotide. That's why it is a single stranded polymer of ribonucleotide. And RNA contain purine bases and pyrimidine bases. Regarding purine bases, they contain both adenine and guanine. But in case of pyrimidine, RNA contains cytosine and uracil, and thymine is absent in RNA. In case of DNA, they contain cytosine and thymine, and uracil is absent in DNA. And the sugar molecule in case of RNA is ribose sugar. In case of DNA, it is deoxyribose sugar. So the sugar molecule is ribose sugar. And the Chargaff rule is not applicable in case of a single-stranded RNA molecule. It is applicable only in case of a double-stranded DNA molecule. As per the Chargaff's rule, the guanine content is equal to that of cytosine and adenine content is equal to the thymine. But in case of RNA molecule, it is not applicable. So in RNA, guanine content is not exactly equal to that of cytosine, and adenine, it is not exactly equal to that of uracil. Coming to the different types of RNA. There are three major types of RNA. One is known as a mRNA or messenger RNA. The second one is tRNA or transfer RNA. And the third one is rRNA or ribosomal RNA. And all these types of RNA having some role in protein synthesis. Before going to the discussion part of different types of RNA, you should know something related to transcription and translation. And all of you know regarding DNA. DNA contains the genetic information, right? So this genetic information produces some effect in our body. To produce their corresponding effect, that genetic information should be converted into some effector protein. But the problem is protein synthesis cannot be immediately directed by the DNA. And this DNA require a intermediate molecule, and that molecule is known as a RNA. So during transcription, the genetic information present in DNA is converted into an RNA molecule. So genetic information is converted into the genetic information of an RNA molecule. That is known as a transcription. And that genetic information present in the RNA molecule is used to produce a new protein molecule. That is known as translation. So during translation, using this RNA molecule, our body produces protein molecules. So these are the two steps which are involved in genetic expression, that is transcription, and second one is translation. During transcription, an RNA molecule is produced from the corresponding DNA molecule, and during translation, that genetic information present in the RNA molecule is used for the production of the corresponding protein. So this is the flow of genetic information in a normal cell. First of all, the genetic information is present in a DNA molecule, and this is converted into an RNA molecule, and that process is known as a transcription. And during translation, the genetic information present in the RNA is converted into the effector protein. That is known as a translation. So coming to different types of RNA. The first one is messenger RNA or mRNA. So similar to all type of RNA, it is a single-stranded structure and it is a polymer of ribonucleotides. So we can say it is a single-stranded polyribonucleotide, and it is produced by the process of transcription. So transcription produces the corresponding mRNA from the DNA molecule, and this mRNA carries this genetic information from the DNA and transfer this genetic information for the protein synthesis. That's why they are known as messenger RNA or mRNA, because they carry the message from DNA molecule. Coming to the structure of mRNA molecule, it is a single stranded structure. So one end is 5'N, the other end is 3'N. So this 5'N, there is a specific chemical molecule that is known as methylated guanine molecule, and this methylated guanine form a cap at the 5'N, and at the 3'N there is a specific molecule, that is multiple adenine bases are present in the 3'N. So 5'N contain methylated guanine and 3'N contain multiple adenine bases. And mRNA, during their immediately after their formation, it is known as immature mRNA or heterogeneous mRNA. So this heterogeneous mRNA contain both coding region and non-coding region, and these coding regions are known as exons and non-coding regions are known as introns. So that's all regarding the structure of mRNA. So 5' end of the mRNA contain methylated guanine residues, and 3' end, that is the tail region, which contain multiple adenine bases. That's why it is known as poly-A tail, polyadenine tail. And they contain both coding region as well as non-coding region. And this structure having specific function, that is 5' cap and polyadenine tail. So the 5' end contain methylated guanine residues. And that this methylated guanine having protective function. They protect 5' end of the mRNA by 5' exonuclease enzyme. So 5' exonuclease is a hydrolytic enzyme that hydrolyzes 5' end of the RNA molecule. So methylated guanine residues present in the 5' end protect this mRNA from 5' exonuclease activity. Similarly, the 3' end contain multiple adenine bases, which also protect this mRNA from 3' exonuclease enzyme. Coming to the second type of RNA, that is known as transfer RNA or tRNA. They transfer amino acid from cytoplasm to the ribosome. So amino acids are present within the cytoplasm, but protein biosynthesis takes place in ribosome. So this tRNA helps in the transport of this amino acid from cytoplasm to the ribosome. That's why they are known as transfer RNA or tRNA. Similar to other types of RNA, this tRNA also having a single strand structure, but there will be significant hydrogen bond interaction within this strand. So this intra-strand hydrogen bond result in the formation of a specific shape for this tRNA molecule. That is, tRNA molecule develop a clover leaf shape. So this is the structure of tRNA molecule, clover leaf shape. That is the secondary structure of tRNA molecule, and it is due to intra-strand hydrogen bond. So this secondary structure of tRNA molecule, it is having 4 arms. One is known as acceptor arm, the second one is known as anticodon arm, and the third one is D arm, and the fourth one is pseudo-uridine arm. So tRNA, it is a single strand molecule. So one end it is a 5' end, and the other end it is a 3' end. And at this 3' end, this tRNA molecule having a specific sequences, which is known as CCA sequence. So this CCA sequence is present at the 3' end of the tRNA molecule, and this 3' end act as a acceptor arm. That is, they accept the incoming amino acid from the cytoplasm. And this anticodon arm read the nucleotide bases present on the mRNA molecule. So actually this tRNA molecule act as an adapter molecule. The anticodon arm of the tRNA read the nucleotide bases present on the mRNA, and at the same time this acceptor arm of the tRNA molecule receive this amino acid from the cytoplasm, which is along with the information that is received from the mRNA molecule. That's why the function of tRNA is considered as an adapter molecule. So the first one is acceptor arm, a highly conserved sequence, that is CCA, which is present at the 3' end of the tRNA. And this 3' end of the tRNA is the acceptor arm. And the second arm is anticodon arm, and which is opposed to the acceptor arm. And this anticodon arm interact with the codon or the bases on the mRNA. So this is the anticodon arm, and this anticodon arm read the sequences present on the mRNA molecule. As per the information received from this mRNA, this acceptor arm receive this their corresponding amino acid molecule. And the third arm is known as D arm, because this D arm contain an unusual base that is known as dihydrouridine base. So unusual base present in the D arm is known as dihydrouridine. And the fourth arm is known as pseudo-uridine arm, because they also contain another unusual base which is known as pseudo-uridine arm. So coming to the functions of D arm and the pseudo-uridine arm. So anticodon arm of the tRNA read this mRNA molecule, and as per the information received from this mRNA, this acceptor arm receive their corresponding amino acid from the cytoplasm. So the amino acid attached to the tRNA through this acceptor arm, and that molecule is known as amino acyl tRNA, that is tRNA containing amino acid. That process is known as charging of tRNA, and that reaction is mediated by a specific enzyme which is known as amino acyl tRNA synthetase. So that is the first part of this translation, that is formation of amino acyl tRNA. And for protein biosynthesis, this tRNA transfer this amino acid into the ribosome. That is the second part. So this amino acid tRNA is transferred, or amino acid tRNA is attached to the ribosomal surface. So there are two functions. One is formation of amino acid tRNA by the help with the help of an enzyme known as amino acid tRNA synthetase, and the second part is the attachment of amino acid tRNA to the ribosome surface for protein biosynthesis. And this DHAM function is they helps in the recognition of tRNA specific amino acid tRNA synthetase. So this dihydrouridine HAM help in the formation of amino acid tRNA. That is the function of D arm of the tRNA. And regarding the function of pseudouridine arm, they are involved in the binding of amino acid tRNA to the ribosomal surface. This is required for protein biosynthesis. So that is the function of pseudouridine arm. Coming to the third type of RNA, that is known as ribosomal RNA or rRNA. So the most abundant RNA is the ribosomal RNA, and they are associated with ribosome, and they have role in translation. So that's all regarding the RNA structure. Thank you.